Welcome, future doctors and NEET aspirants. SuperScores SS Empower Your Dream presents a high-octane masterclass on one of the most brilliant engineering marvels in the entire human body, the kidney. I'm so thrilled to be your guide for this explainer today. We're going to break down the master filter, its form, its intricate functions, and ultimately what happens when it fails. If you want to secure those top ranks, mastering this topic is absolutely non-negotiable. So let's dive right in and get started.
Here is our roadmap for today. 1. Gross anatomy. 2. Internal macrostructure. 3. The nephron unit. 4. Renal physiology. And 5. Clinical signs of dysfunction.
Section 1. Gross anatomy and location.
Notice the Morris quadrilateral right here. This is a highly testable fact for your exams. The kidneys actually lie inside the specific parallelogram, aligning exactly with the 11th thoracic vertebra at the top and the 3rd lumbar vertebrae at the bottom, so T11 to L3. You need to lock that spatial positioning into your memory right now.
Now, it's also crucial to really understand the 3D anatomical context. The kidneys don't just, float around freely in your belly. They have what we call a retroperitoneal location. That basically means they sit entirely behind the peritoneum of the abdominal cavity. Plus, they're wrapped up in a protective fibrous capsule and supported by tough fascial sheaths. It's essentially heavy-duty packaging that keeps them secure and shielded from any physical trauma.
Section 2. Internal macrostructure.
If you were to slice the kidney open, two very distinct zones emerge. You've got the lighter outer renal cortex and then the darker inner renal medulla. See those triangular fan-like structures sitting in the medulla? Those are the renal pyramids. They are the absolute core structures where the heavy lifting of urine transport actually begins.
So the crucial point here is the pathway of drainage, because once urine is formed, it doesn't just magically teleport down to the bladder. It follows a very strict rapid-fire sequence. It drains from the apices, or the very tips of those pyramids, which we call the renal papillae. From there, it flows into a minor calyx, and keep in mind, each minor calyx gets fluid from about 1 to 3 papilla. The minor calyces then merge into a major calyx, which finally empties right into the funnel-like renal pelvis. Memorize this exact sequence: papillae, minor calyx, major calyx, renal pelvis. It's a classic NEET question just waiting to happen.
Section 3. The nephron unit.
Take a look at this incredibly beautiful capillary network. A single kidney actually contains over 1 million of these nephrons. Let's break down the primary components. It all starts up at the renal corpuscle, which includes the glomerulus and Bowman's capsule. From there, the fluid enters the proximal convoluted tubule, dives way down into the U-shaped loop of Henle, comes back up into the distal convoluted tubule, and finally dumps right into the collecting duct.
But here's a massive high-yield exam topic you really can't miss. Not all nephrons are created equal. We have cortical nephrons, which have their renal corpuscles sitting in the outer cortex and feature a very short loop of Henle. Then, we have the juxtamedullary nephrons. These have their corpuscles situated deep down in the cortex, right up next to the medulla, and they boast a distinctly long loop of Henle.
You can see the stark difference in depth right here. The cortical nephron's loop barely even dips into the medullary zone. But look at the juxtamedullary nephron. That loop plunges incredibly deep into the medulla. This massive difference in depth is exactly what allows the kidney to create highly concentrated urine.
Section 4. Renal physiology and filtration. The whole physiological process kicks off with ultrafiltration. Think of this as the body's ultimate pressure washing system. Unfiltered blood is vigorously pushed through the glomerulus, and fluid is literally forced into Bowman's capsule. But what's driving this? Well, the physics of it is completely governed by Starling forces, a delicate, continuous balance of hydrostatic and oncotic pressures acting across the glomerular basement membrane. This physical principle is the true engine of kidney function.
If we focus on the vascular pole of the renal corpuscle, this is where the magic really starts. You can clearly see unfiltered blood rushing in through the wider afferent arteriole. It swirls all through the glomerular capillaries, gets filtered out, and then the remaining blood exits via the noticeably narrower efferent arteriole. That specific difference in vessel diameter is exactly what builds up the hydrostatic pressure you need for ultrafiltration.
Once that filtrate is captured, it goes on quite a journey. Step 1 is the proximal convoluted tubule, or PCT. The cells here have a massive brush border made of long microvilli, increasing the surface area so drastically that an incredible 65% of the filtrate is obligatorily reabsorbed right here. Step 2 is the loop of Henle, where the thin descending and thick ascending limbs work together to concentrate the urine. Finally, step 3 is the distal convoluted tubule, which handles further transit and fine-tuning back up in the cortex, often under the direct influence of hormones like ADH for final water reabsorption.
Let's visually trace this pathway. From the swollen Bowman's capsule, heading down the descending limb, looping right around the hairpin turn of the loop of Henle, back up the ascending limb, and out to the collecting duct. Locking in this visual pathway is going to make answering sequence questions on your exam an absolute breeze.
Before we wrap up physiology, we absolutely have to look at this crucial anatomical intersection, the juxtaglomerular complex. Notice how the distal tubule comes right back to nestle between the afferent and efferent arterioles. You have the macula densa cells of the tubule and the granular juxtaglomerular cells of the arterioles right there. Together, this complex is absolutely vital for your blood pressure regulation. It constantly monitors sodium levels and can trigger the renin-angiotensin hormone cascade to keep your systemic pressure perfectly in check.
Section 5. Clinical signs of dysfunction.
Now, the National Kidney Foundation outlines 10 key signs of kidney disease, and we can directly link them back to the physiology we just learned. For instance, foamy urine? That happens when the glomerular filtration barrier fails and starts leaking structural proteins into the urine. Swelling, or edema? That occurs when the nephrons fail to properly balance fluid and sodium reabsorption, causing water to pool up in the tissues. And unexplained fatigue is often tied to the kidney's failure to produce hormones like erythropoietin, which is essential for red blood cell production.
So, I want to leave you with a provocative question to really think about. How does mastering the structure of this master filter today empower you to spot these clinical signs early on as a future doctor?
The honest truth is, every single symptom your future patients describe will trace back to a failure in the very tubules and capillary beds we just explored. Your exam prep today is literally saving lives tomorrow. And that brings us to the end of our explainer. Keep revising that gross anatomy, visualize those nephron loops, and remember your Starling forces. You totally have what it takes to crush this exam. This has been Superscores SS. Empower your dream. Keep studying hard, and good luck out there.